Marianne Martone, who will be the moderator for our first session here.
Thank you very much, Emily. It is my great pleasure to moderate session 3, which is called Best Practices in Data Sharing. This session comprises 2 themes that are near and dear to my heart. First, we will be hearing from Karen Yook, who had the Making Data Usable project. Karen is editor and database curator at Caltech with microPublications and also WormBase. And she is really going to be highlighting the importance of curation in effective data sharing and how to incorporate that into the publishing workflow. Then we have Kristen Briney, who is a biological engineering librarian, also at Caltech. Her project name was Making Data Usable, and she's going to be talking about a very simple but extraordinarily effective practice, which is naming data files. And this one of the questions that I get all the time in data management from researchers is, what exactly am I supposed to do? And I think both of these presentations really highlight effective strategies. So without further ado, let me introduce Karen, who's going to be talking about making data usable while publishing.
Thank you, Marianne, and thank you to FASEB for seeing my project. I'm going to tell you about a project that we started that stems out of WormBase curation. WormBase is this is microPublication biology. This is an online open access journal. We publish single peer-reviewed single result articles, single figure articles, and these publications are receive PMIDs or DOIs. They're available through PubMed and Google Scholar. And really what I'm going to focus on is how we're moving data directly from published articles into curated databases, such as WormBase, Saccharomyces genome database, and FlyBase. This journal is published by the Caltech Library, and you'll hear from Kristen Briney, not on this project, but on our own things, and she is a key to our publishing team. Okay, this is the actual journal team. What really Paul Sternberg is our fearless leader. Tim Shadle from WashU is also key to guiding us. He's one of our senior editors. And let me introduce you to Danielle Ravachidi and myself. We are managing editors. Nick Stifler is our very active developer. And I will be highlighting some work by Valerio Arnaboldi, who has created our curation tools that we have embedded into our publishing platform. So one thing to really point out is that we're all from WormBase. So we started as WormBase curators, and what we noticed, in thinking about fair data and just curation in general, is that the database curators are the ones who actually ensure that data are fair. Things can be published and they can be findable through PubMed, but what actually makes things fair? And I will go over a couple examples of how we do this and when this fails, what happens. Not curation fails, but when people publish data, how we can make that fair and what some kind of problems that come along the way. So we curate data from peer-reviewed journals. This includes paywall journals and supplemental files. This data is often kind of hidden behind these file types, and also behind, they're not accessible to everybody. We're a model organism database. WormBase is the major knowledge base for the model organism Synovitis elegans, and it's open to all. Alliance of Genome Resources is also a conglomeration of the model organism databases and takes data from us. That's also open from all, and you can peruse very relevant biomedical organismal data there. How do we make these fair is that we take biological entities that are published in these peer-reviewed journals, and we annotate them, we extract them, and we assign unique and persistent IDs to the different objects, the entities. And we also do a number of things that, in thinking about reusability, it's the database curators that are actually establishing a lot of rules that make these data reusable. These include establishing nomenclature rules for the community, assigning IDs, recording critical details of the biological entities that are used in experiments, such as the composition of these entities, how they're generated, and who made them. So it gives provenance to what these entities are and who to go to if you need more information or who to get them from. In the end, sorry, I should be clicking along the way. So in the end, what happens is that we've actually been aggregating community work through annotating these entities. And this also, if you can see, it's such a, one entity, this transgene, what we do is we aggregate all those papers that use that entity. So there's a huge record of these entities that are being created by our community members, and we are cataloging them and having them openly accessible to people. Another thing that is part of the reusability criteria is that data usage licenses are known, but these are really established by the journal. So let me show you some examples of how what we do is really important. So looking at genetics papers in 2017, genetics in G3, papers published by the GSA, just a survey of 68 papers. As a curator, 68 worm papers. As a curator, I found that 36.8%, 25 out of 68 of the papers have nomenclature errors. That means that they're, the authors are reporting data and entities that do not conform to the worm community nomenclature rules. There might be typos, there might be, they range in severity of how they would actually affect downstream reusage of their work. And we can, just formatting errors alone. I think I need to explain that WormBase nomenclature rules include very strict formatting rules, such as capitalization of certain entities and not other entities, and also italicization is a very key thing. So when people don't italicize things, they're not going to be found so easily. So, but these might seem minor, but there are actually, looking very carefully at these nomenclature issues, there's 10% which actually are critical, and they really affect how people are going to reuse the data. So, for example, in this fly paper and in this worm paper, you don't need to read these little snippets, but these are curator comments. In this case, the author was trying to use using a wrong short name for the gene that they were studying. They wrote the paper using this short name, but the short name actually is a different gene altogether in FlyBase. So that's really critical. In this one, and the one on the right, the author was using the wrong name construction convention for reagents built in their lab. These are not going to be curated into the database. They're not going to be findable. They're going to be very hard to pass along. So these really do, having a curator look at these things, we find them. So other things that we find that affect usability is illustrated here. And this is really bad because it's been propagated to different papers. So in this first instance, all you have to do is look at the little blocks of what constitutes a strain, the entity, and this is the genotype. What happened is the entity name got swapped. So this strain in this first paper, stands for this object, but in the next paper, they switched these two entity names. So, how can you tell what they're actually looking at? These are reporters. These reporters for specific genes. One is NPHP1, the other one is NPHP4. They actually have very different expression patterns. So depending on who they ask for the strain, they're going to get different things. Is that going to result in, what's that going to result in people's research? Another big error we find is that authors just omit things. In this case, author left out the source of the strain and critical details of the reporter construct, such as what was used to find and define the region that's being looked at in the gene. And now without this information, how can people evaluate the work, let alone reproduce it? So it's very critical to have curation within a pipeline, a publication pipeline, to catch these errors and correct them before publication. And that's what we've done. So micropublication biology, with this journal, we aim to publish reusable data. And what that means is that we actually put curators as part of the publication process. And now what I'm going to do is explain what curation means and how we've embedded it into our pipeline. First, there's entity identification, extraction, and validation. What this means is that if you just look at a piece of text, can you tell what the entities are, and can we extract them and validate them? So these are the entities. If we highlight them, how do we find them? The other part of curation is fact extraction. So can we build tools that will help us really extract what the actual annotation should be? So in the first entity extraction, we identify 2 entities, DAF18 and an allele, SY887. Those details don't matter, but just to know that they're biological entities. And in the second part, we need to write out that we need to annotate that this allele of DAF18 causes a phenotype. In this case, it's brinjal pumpkin. So again, looking at a paper, this is a paper in micropublication in our portal. This is what it looks like. What we can do is we run an entity identifier, and we can color code things based on known objects. So in this case, DAF18. On the right, we see a list of all the objects that were pulled out from our identity and identifier. And we also have things in green that are predicted based on the fact that, again, in a lot of the model organism databases or a lot of model organism communities, there are very strict nomenclature rules. So those nomenclature rules can be used in automated recognition to find and predict these objects. And by doing this, we can also see what objects are predicted, but are they also typos? Are they known? In which case we can get back to the authors and say, fix it. Or are they absolutely new articles? So in this entities, and in this case, these are new, so we can curate them right away. So we add them to the database. We can identify all sorts of different classes of entities. In this case, WormBase, we identify quite a few. So what does this look like when we identify an object, we link it, and we can also edit it. So if it's the wrong link, or we can get a little more information, and we have and we can embed a link directly to the database. So this very much speeds identification and exploration of more biology about this gene for the readers. But also, as I mentioned, things get predicted and we can create them in the database right away. So this is very important because, and again, if you don't have the right entity, in this case, this is a sentence from a paper that came in, and the author named these named alleles of these 2 genes, XOC8 and RLB1. It was just predicted. So I knew right away that, oh, this is predicted. Let's go see if it already exists and if it's a typo. And in fact, it was. So it should be okay, 2523. In this case, this is a very serious error in that they named an allele, but it was actually an allele of a totally different gene. So our, having a curator embedded in the system allows us to find and get back to the author and say, this is important, you need to change this. So this is the other flip side of nomenclature identification and adhering to rules. Any nomenclature error is going to affect AI. So that's just something to keep in mind as we go through. So let's go on to the second part in fact extraction. So what we need to do is what we want to curate is that this knockout of a gene has a higher frontal pumping rate. So we require more than just identity identification. As I mentioned, Valerio in the very beginning, he helped us build in curation forums into our system. So we have everything that's found in through our entity extraction and identification gets pre-populates these drop-down choice lists where we can just click through and create an annotation. And anything that's missing, we can add. So these also embed ontologies, so they're controlled vocabularies to make sure that we are all using the correct terms that are also computer friendly. And in the end, we have an annotation that we can send directly to WormBase upon publishing. Okay, so this is last slide. Micropublication authors' data are licensed and archived in now Caltech data. So this is the last criteria for reproducibility. We give CC by 4 licensing to the articles, which means that you can use it, really usable, but you have to cite it. So again, preserving provenance, and everything is archived in Caltech data. So this leads back to the beauty of our library collaboration at Caltech. And with that, thank you very much. Thank you for having me, and thank you for seeing my work, our work.
Thank you very much, Karen. If you have any questions, please type them into the Q&A function, I guess we would call it, and we will get to them at the end of the presentations. So now I would like to introduce Kristen Briney, who is going to be talking about naming data files descriptively for easier reuse.
So thank you very much. I'm excited to be here. As mentioned, my name is Kristen Briney. I'm the biology librarian at Caltech, and we're going to be talking about file naming conventions. Before I get into my talk, I do want to recognize that I'm representing the Caltech Library today, and the other half of my team is unable to be here, Tom Morell. He is the photo on the left. The photo on the right is our main library branch, Sherman Fairchild Library. It's a lovely 4-story building that I work in. I also want to recognize the other winners of the prize because I'm very, very impressed with the amount of detail and infrastructure that has gone into those submissions and that work, and that's very necessary and critical work for data sharing and reuse. And what I'm going to be talking about today is not on that scale at all. I'm going to be talking about something very small that you can do by yourself to make data reuse easier. So this is something that's going to benefit you, first and foremost, your future self, and maybe your lab mates, and then definitely your collaborators, and probably people who are going to be using your data if you post it publicly. So what I'm talking about can best be summed up by this internet meme. It's one of my favorites. So in this meme, we have Gandalf the Grey. It's from the first Lord of the Rings movie. And in the movie, he's going through the mines of Moria. But in the meme, he is checking his raw data folders, and he's looking around, and he's thinking, and he's very confused. And he says, I have no memory of this place. And I feel it really encapsulates something that happens to a lot of researchers, myself included, that we do research for a certain amount of time, and then we go back and try to find our files from before, and we can't find them, or we find them, but we're not sure exactly which file that we need. And it's very confusing, and our memory's not very good. I can hardly remember sometimes what happened last month, let alone last year. And if I have to find my old data files, it's very difficult. So we need some way to make this process easier. And one of those ways that I like is file naming conventions. So these basically act as guideposts or street signs or labels on a map. So when you're navigating through all your files, your new files, your old files, you can figure out what they are. Because the truth is, as people, as researchers, we lose our own files, and it's even worse with other people's data. It's very difficult to get a group of files and try to figure out what's in them without any sort of information. So we need good labels, and we need those labels to be separate from our lab notebook, because that might be available, might not be available. We may take very bad notes, and probably also separate from repository metadata when we're sharing data, because it's possible that those files got moved on your computer. And what are these files again? I'm not really sure. Where was the record? I don't know. We need the files to label themselves so we know what they are, so we can reuse them. So let's talk about what a file naming convention actually is. So a file naming convention is a way to richly describe a file containing lots of information or metadata. And file naming conventions put related files with consistent related patterns of names, which helps you organize your files. And then finally, they provide enough information to disambiguate related files. So I collected these two files from the same source. How are they different? Well, it's in the file name because I put enough information in there for me to tell. Why should you use a file name? If you haven't discovered these already, let me tell you why they're the best. So file naming conventions make it easier for you to go back to your old files and find what you need. And that searching can happen visually because you can just scan through your file names and look at them. You can also do system searches because that metadata or information is in the file name itself. You can actually search for that information. And best of all, you don't actually have to open your files to figure out what's inside because they're labeled well. The computer scientists in the room probably know that consistent file naming makes files more computable. So not only can you consistently label the outputs of your computer program, but you can label the inputs and say, harvest everything with this pattern in the name and go. It makes it a lot easier. And file naming conventions are flexible. There's not one file naming convention to rule them all. Sorry, another Lord of the Rings reference there. But you basically can create file naming conventions for different groups of files, different projects. Every time I start a new project, I create a new naming convention and put whatever I need in that convention for that project. Finally, if you're going to label your files, it will save you time later. That means if you can't find your file, you might have to recollect. Normally, you might have to recollect that data. But if you can't find it, that saves you a huge amount of headache to actually have to go back and recollect data that you already have somewhere, maybe on your computer. So let's look at a couple examples of file naming conventions. These are actual screenshots from my computer. So the first one is my journal PDF. So these are articles I've downloaded from my computer, and I use the file naming convention first author last name _ 4-digit year _ abbreviated title. And I like this convention because it helps me associate the citation with the name of the article. So I can say Cummings 2020, and that's for assessing research compliance. And it really helps me in my brain to see those associations. Also, it really is organized really nicely by author name. So I can just put my finger exactly on, oh, I need that Davis paper right there. It takes me about 5 seconds to rename my article PDFs when I download them, but I always know exactly what I have and I can find exactly what I need. And if I can't find it, I didn't download it. The other example I want to show you is my presentations folder. So I teach a whole variety of things. I teach for classes, I teach for freshmen, I teach a whole bunch of things. And so I have a whole folder of presentation materials organized by date. So I use 4-digit year - 2-digit month - 2-digit day _ the event title. So when I do this, I can basically say, when was the last time I taught Data Visualization 101, and just scan up and use that PowerPoint and copy it into this time I'm going to teach Data Visualization 101. And it makes it a lot easier to keep track of what I've taught and find the files I need for the next time I teach that topic. Okay, so I'm an instruction librarian, and I'm actually going to teach you right now how to name files. So this is actually the recipe that we won the award for. It's the file naming convention worksheet. So for the rest of my time, I'm actually going to walk through the 8 steps of the worksheet. And I encourage you to name some files with me, or at least follow along and figure out how it works so you can name some files later. So you're welcome to follow the link I put in the chat. You are welcome to scan the QR code and get there, or you can follow the bit.ly at bit.ly/file naming worksheet, no spaces, and the capital of first letter of each word is capitalized. So F in file, N in naming, and W in worksheet. It's actually important for the shortened link to put those capitals in, otherwise you won't find the worksheet. So pull up the worksheet or follow along. I just want you all to know how to do this because I really think they are some of the best, easiest things you can do to make data management and data reuse easier. Okay, so let's get started and start with step 1. So the first thing you want to do is figure out what group of files you are going to create the naming convention for. So in the example, oh, sorry. Yes, my mug says, show me the data. It's very appropriate for today. So the example I'm going to be working through is for a group of microscopy files, and I'm going to name everything from the raw image all the way up through the processed image. So figure out what group of files, and it doesn't have to be all your files, just a group of related files you're going to create a convention for. Step 2, what information or metadata is important about these files and makes each distinct? What do you need to know at a glance to understand what's in a file and to tell it from its neighbors? I like to recommend about 3 to 5 pieces of information to keep the file name size reasonable. So for the images, I'm going to use date, sample ID, and image number for that sample on that date. Step 3, what do you need to do to make some of that information a little bit shorter and smaller in size? Can you encode it? Can you abbreviate it? So instead of writing the full title of an article, can you write a short title? For example, instead of saying the whole Latin name of a species, can you abbreviate it in 2 or 3 letters and make that human readable, but also short to keep the file names from being unwieldy? So in my example, we're going to use a code for sample ID, and it's going to have 3 components, a 2-letter project abbreviation, P1, P2, for lack of creativity, a 3-letter species abbreviation, mouse is MUS, fruit fly is DRS, and a 3-digit sample ID, so it can have up to 999 samples. Step 4, what order are you going to put those 3 to 5 pieces of metadata in? And order matters mostly for how you're going to organize things on your computer and how things are going to sort. So the most important piece of information should go first, and then kind of a hierarchy from there. So however you think about organizing that information in your head, use that to determine the order. So for my microscopy images, we're going to start with sample ID first, because that's kind of the reference I use for cross-referencing everything, followed by date and then image number. Okay, I'm going to have a brief aside here. I'm going to pull out my soapbox to talk about ISO 8601. I love ISO 8601. I believe there's one proper way to write a date, and I cannot talk about data management without talking about ISO 8601. So thankfully, XKCD agrees with me. This is XKCD Comic 1179. And the proper way is to write a 4-digit year dash 2-digit month dash 2-digit day. And you saw that earlier when I showed you the picture of my folders and how they are organized. So there's a couple reasons for using the standard. The first one is, say, let's have a meeting on 7/12/2024. I'm in the US, so obviously I mean July 12th. But if I were sending this date to somebody in the UK, they're like, why do you want to have a meeting in December 7th? Because US and UK write their dates with different formatting and different order for the numbers. So having a little clarity really matters. The other reason I like ISO 8601 is because when you use it, all your dates sort chronologically. We saw this in the example I showed you, but you basically have your biggest unit of time, your medium unit of time, followed by your smallest unit of time, which makes everything sort. So this works really well in file naming conventions, and you can always use it for your data as well and use it in your data. Do note that the standard, you can use it with or without the dashes. I tend to find the dashes, using the dashes to be more human readable to kind of visually separate everything. But if your file names are getting long, throw the dashes out and make your file names just a little bit shorter. Okay, I'm putting my soapbox away. Let's go back to step 5. So we have figured out a group of files. We've figured out what information is important about those to put in the file name. We've shortened some of that information, and we've ordered that information. Now we want to kind of segment those pieces of information and make them visually separate, and that will help with computation as well. So I tend to use underscores and dashes to separate my pieces of information. I am firmly in the camp of no spaces in file names, please. You only have to use the command line a few times with file names that have spaces in them to absolutely hate spaces in file names. So it's just better to not have spaces. The point of view that I really like for using underscores versus dashes is to use underscores to separate unrelated pieces of information and to use dashes to separate related pieces of information. So I'm going to use underscores in my example to separate my big pieces of metadata and dashes between those 3 parts of my sample ID. All right, There's only 3 more steps to go. So step 6 is to track different versions of your file. And usually I like to do this on the end of the file name. So we have underscore v1, underscore v2, underscore v3, never underscore final. Final's never actually final. You could do underscore the date. In this case, I like to do, for the microscopy example, underscore raw, underscore processed, underscore composite to tell the type. And that way you have the same root of the file name with a little information on the end to say how it's different. So basically, I've done this with interview data, and you have the same data set, but now you have underscore audio, underscore video, underscore transcript, underscore notes. So you can have the same root of the file name. So this is obviously still the same data. It's just in these 4 different formats. So putting that version information on the end can be really helpful. And this is how I write manuscripts. I just save new versions occasionally. All right, last 2 steps. Let's write this convention down because we've worked everything out. So when I write my conventions down, I like to write out both the pattern as well as an example. So let me tell you an example of the convention we just made. It's P1-MUS-023 underscore 2020-0229 underscore 51 underscore raw. So this is project 1, mouse number 23. The image was taken on leap day of 2020, image number 51, and this is the image that came off the microscope. So I just told you a whole bunch of information about that image from this maybe like 20 to 30 character file name. So we have a lot of information there, and I can tell you exactly what's in that data without any other info. I don't need my notes. I don't need a repository record. It's just right there in the file name. And if I tell somebody else my pattern, I can give them the file and they'll know what's in my name too. All right, last step of the worksheet is to document your convention in a README file, or print out the worksheet and paste it next to your computer, or write it at the front of your lab notebook. Basically keep it with your files. And that way, when you come back to your files in 6 months, you have a document that says how to interpret the file names that you use. Okay. Hopefully, I've convinced you that file name conventions are one of the best things that you can do to help reuse your data, that really there's something small that have a big payoff. They save you time and headache when you're trying to find files later. So oftentimes, like, I will find my files in 5 seconds because I've named them appropriately. They help when you don't have your lab notebooks on hand to figure out which file is which. Well, it's right there in the name. They prevent you from recollecting misplaced data, and that is important. I've had to recollect data that's already been collected, and nobody enjoys that. Nobody likes that. And best of all, file naming conventions are free. They don't cost you any money. It takes maybe 10 minutes, 5-10 minutes to figure out your naming convention at the start of a project, and then 5 seconds every time you name a file. But that pays off in hours when you're trying to find things later. Last thing I want to say is that the file naming convention got added to a workbook that Caltech Library put together and put out last fall, full of data management exercises, everything from organizing your data to determining who has rights to what data, to a checklist for sharing. And I just put up a checklist for making your spreadsheets more accessible or reusable. So do check that out. Everything in the workbook is under a Creative Commons attribution non-commercial license, so you can reuse it, you can edit it and modify it however you want. Just put a citation on there and don't sell it. That's pretty much it. So with that, I hope you've learned a little bit about file names. I hope I've convinced some of you to start using file naming conventions. I'm happy to answer questions, and I really appreciate the opportunity to talk to you, and appreciate FASEB for the prize. Thank you.
Thank you very much, Kristen and Karen, both for excellent presentations. If you have questions, please put them in the Q&A button, and I will get to them. While I'm waiting for the questions to come through, I just wanted to ask a question of both our speakers. I'll phrase it a little bit differently, which really deals with generalizability. So Kristen, since you went last, I'll ask you first. In a workshop from the National Academies on data, changing the culture around data management and sharing, they really emphasized that the lab was the unit of data sharing, right? Because you really do need to share data within a lab. And one of the big problems, of course, that PIs have is that as people leave, each individual has a way of doing things for themselves, but it doesn't generalize. And so you are left with things that are difficult. So it seems like the file naming convention could easily be implemented on a lab-wide basis. And I just wanted to know your experience with that or any advice that you might have.
Yeah, certainly. So
Mhm.
I take a somewhat selfish view of data management, that I'm the person most likely to reuse my own files. And so I think that we should be doing data management to make it easier for our future selves. And then the lab is important too. But I also think that the more you're sharing data actively during a collaboration, the more important it is to name your files jointly. So I did a project, we had 8 different universities collecting hundreds of files at each site and contributing them. And the only way that we kept anything organized was we had a very strict file naming convention for all the files, and anybody could find anybody else's files during the project, after the project, but everything stayed organized because we named things appropriately. So I really think that file naming conventions can make the difference between using files from other people and sharing files. In that case, I really think file naming conventions are almost required for really collaborative research. That said, I think that as long as you name your files accurately and descriptively and document your convention and share your convention, like if I have a convention, I write down how it works and I share my files with somebody else, they should be able to understand it. So I care a little bit less about making sure everyone uses the same file naming convention if you're not sharing files actively and working together on a project. As long as you are using a convention, you're documenting things, and it's descriptive enough. So I'm kind of, whatever works best, I like to kind of customize to whatever needs to happen in the lab to maximize reuse. But there's definitely a lot of things that need to be done to make sure data is reusable within a lab. And there's a lot of, I'm not going to talk about all of them. I can talk about, but there's a lot of work to be done.
Thank you. And Karen, I am old enough to have been to be around from the very early days of 4th11 and other groups who really sought to transform the way that we publish in science. And the micropublication, the nanopublication has been, the idea has been around for a very long time. And it has been traditionally very difficult to get uptake of that concept, this sort of single figure or a single result post that is done in a way that allows it to be imported into databases and other structures so that it can be used. And you clearly have had some success here, and actually a lot of success, which I think is great. How much do you think depends on the sort of nature of your community, that you have, WormBase, you have a smaller group that's sort of committed to working in this way, versus broader expansion into the scientific community? Would you be able to comment on that?
Oh, absolutely. This is such a key thing and a key element of, I think, why we're successful. Because our community is, it's small. It started, in the worm community, and we've been able to branch out. So we basically proved that this was useful, and it's useful in a general nature for, like all labs, whatever you're working on, because it's single figure, and we don't need a complete story. And you just need to have a good experiment, and then we work with the authors to make it, really robust in terms of fairness, right? So it's which doesn't necessarily translate to other communities, because they don't have nomenclature rules, right? There's a lot of similar communities don't even have databases. So what are we going to do with those? But I absolutely, you totally nailed it, Mary. And I think it's because we started from the community up, and we started with that need within the community. And actually we started even smaller, because we started within the WormBase database community. We're like, we can't keep up with curation. So we're going to need to train authors to curate. And that's the next step that I didn't really talk about, is that these tools, we're making them simple enough for authors to use them. So we're going to be rolling out the author linking that they can, it's basically a bioentity spell check for them. And that's, hopefully authors will find that cool. And as we expand, we've already, gotten a lot of different organism papers. As we expand, hopefully we can also start bringing in other nomenclature rules and, have a more holistic biological, spell checker across all realms of biology. But the community first, and we just kind of, moved on from there. But the other thing is it expanded. It's really been expanding by word of mouth from people within universities, especially small colleges and non-R01 funded colleges. So the small PUIs, they're doing all this research, and they have a venue to put it out there. So it's really just been phenomenal looking, and I think it's spreading that way now. So now we're seeing, not just organism-based communities, but we're seeing, primary undergraduate institutions, that community. So we the discussion of what a community is, in terms of publishing needs are changing for us. I mean, our perspective. Oh, I can't hear you, Marianne.
It's one of the reasons why it's so important that these types of stories get out there, because you clearly show that, first of all, you work out both the infrastructure that's required, but I think more importantly, the human infrastructure as well. And as I mentioned in the beginning, curation is one of the topics near and dear to my heart, because I really feel that curators in this early stage of data sharing are a critical piece to really help the researcher and things that are not natural to them. It's not natural of them to follow these nomenclatures rigidly or deal with ontologies, and curators can help smooth that out, thereby again providing many benefits, including for AI. But I think the interesting thing about these infrastructures, and that leads us into the question that's in the Q&A, is that, right, there's many ways to solve these infrastructures. You don't need to be bound to one thing. And so we do have a question that says, why did you choose to use CalData versus Figshare or some other generalist repository?
I'm trying to figure out who actually should answer that, because both Karen and I work on this project together.
Yes. Yeah.
I can defer to you about the specifics, but it just was a natural fit. Caltech the Caltech Library was our publisher, or is our publisher, and they know how to archive data, and they do it for Caltech professors, they do it for Caltech researchers. So they already have an archive system, and the archive system has a DOI, and that was the bar of like, how do you a good practice for archiving data, right? That DOI, and they already have it. And our next step is also to start employing Kristen's file sharing conventions for the extended data we have. I mean, the other thing about micropublication is we don't encourage supplemental data, because that should be the paper. All the data needs to be out. So we don't have that much, we don't usually have that much need for it, but we're finding more uses for it. And Kristen, if you want, please add more.
Yeah. So from the institutional perspective, I think it makes sense for us to have a data repository, because there are a lot of subject repositories, but it doesn't cover the whole landscape. And there are the generalist repositories, but we have the ability to do a lot more custom work with our community. And that micropublication is a great example of that, because we can build workflows with them to help pre-populate some of the data they're collecting from the article end and put that into the repository. So I think this is a great example of why a institutional repository actually is a big benefit, because we meet monthly to talk about some of these things, and a lot of months we're talking about how to streamline data set integration into the repository, and just that close collaboration can make all the difference in terms of efficiency, effectiveness, and being able to respond in a way that generalist repository can't.
Mm-hmm. Thank you. And I also think that it's an important point that your project started in 2016. And at that time, you will look around for the infrastructure that's available. And I think one of the important things that has started to happen, you see it with the NIH data sharing policy, is they've started to articulate desirable features. And this is going on across the ecosystem, which back in 2016, we didn't necessarily have these. And so basically, there's a certain set of criteria that you have, and at any given point, you should be able to go and get the infrastructure components that work best in your circumstances. And they may change. If you started this project, if you were doing it today, you might have made different choices. I don't think so, because it sounds like that the institutional repository was important. But I think that that's, again, in terms of generalizability, just an important point that that you should be able to do these workflows across whatever pieces make sense for you as we start to articulate what these pieces should do. So I see Emily is on, and I think we're 1 minute short, but I want to both congratulate the speakers for their successful entries into the DataWorks competition and also for their excellent presentations. So thank you very much, and thank you to the audience too for listening.